Okay, so today, at the beginning of this lecture, we discussed a lot the impact of dispersal and gene flow on the evolution of species niche and species range. But in the two remaining lectures, what we'll discuss is how those dispersal abilities that affect gene flow and migration, demographic migration through space, evolve, and what we understand about the evolution of those dispersal characters. So about more than 10 years ago, I wrote this review that is my most cited paper, but I don't take much pride of it because I think it is cited only because I stole a cool title from Bob Dylan, who since then won a Nobel Prize, but I didn't. And initially, this lecture I prepared for some other course where the command was that I was supposed to say what happened most recently in the field of dispersal evolution. So I'd use that review, try to explain new stuff that occurred within this 10 years since I wrote it. So I'm going to do a little bit of that during this lecture, but also tell you about all the stuff because I think that it's still important and that we need to build on that to understand dispersal evolution. So this review was organized in a very lazy way that worked very well, that I just picked up 10 questions about the evolution of dispersal and then wrote a little nice section about every question. And it's still quite handy to try to discuss those different issues, so I will use those questions to progress within the lecture as well. So the first question is simply, what is dispersal? And there's actually, I'm cheating here because there are two questions. What is dispersal and why is it important? So first, what is dispersal? I must say the main reason people cite this paper is because I provided a definition of dispersal. So then they just use that single sentence to, define dispersal. So it was not an invention from me, it was already in the literature before. So you can describe dispersal as any movement of individuals or things that are not clearly individuals, such as the pollen grain or propagules, with potential consequences for gene flow across space. So you could have different types of dispersal. You could have natal dispersal or breeding dispersal. So you know what natal dispersal is? Yeah, away from your birthplace. It's when an individual born somewhere decides, or not decides, but disperses to some other place and will reproduce in some other place than the place where this individual was born. So that has consequences for gene flow because obviously its parent reproduced somewhere else than this individual will reproduce. So the genes are moving across space with consequences for the evolution of those populations. Breeding dispersal, you know what it is? Yeah, so you've already bred somewhere and you're moving to breed somewhere else. So this has also consequences for gene flow to space because you spread your genes potentially in some places and you're gonna potentially reproduce and spread your genes in some other places. So there are many other types of movement which, according to this definition, are not really dispersal. And sometimes it's not always very easy to distinguish other type of movement and dispersal. So for instance, some animal foraging for food will move through space, but if it forages and come back to the same place to reproduce, for instance, this is not really dispersal in the sense of this definition. So you can explore space to find prey and come back, and in for the same argument, seasonal migration, where animals migrate huge distances from wintering to breeding grounds, but coming back potentially to the same place to breed every year, it is not dispersal according to this definition. But it's movement, and it has also implication, but I will not discuss that so much. Sometimes people discuss it together, like every movement. I will focus more on this type of movement that have consequences potentially for gene flow. So another part of what is dispersal. So dispersal is a movement. Okay, but this movement is a result of many things happening. And some people increasingly find it useful to distinguish different stages of this movement to try to understand better the whole evolution of an ecology of dispersal. So for such a movement to occur, you must have departure of some individuals from some place. You have a stage of transience where these individuals are moving across the landscape. And then you have a decision, or a decision in the broad sense. You have a stage of settlement where this movement will stop and the individual will end up in some new place. And a lot of the literature on dispersal evolution has focused on explaining departure. Why do you leave and why do you stay? But actually has a bit neglected what will happen in the transient phase, and also other type of decisions or effect on dispersal that are due to settlement. When do you stop moving, and why do you decide to stop the movement in such a It's a decision, but I'm talking also about dispersal of things that are dispersed passively by wind or by water or by other animals, because in any of those stages, there might be traits that facilitate departure, transience, or settlement in different types. You also have very different vision about dispersal in terms of describing this movement. So one vision that has been very popular is to imagine space or habitat made of discrete patches. And then what you generally measure is whether individuals depart from that patch or not. So for instance, you will distinguish Kilpatrick individuals that stay in the patch where they were born, and dispersing individuals that immigrate to some other patch after the birth. You will also distinguish like discrete categories, such as resident or immigrant individuals within a population. And often, if you've got this kind of vision of what is dispersal, you are trying to quantify it by dispersal or immigration rates. But you can already see then it's a bit complicated, because what you will define as a disperser or a Kilpatrick will depend on the spatial scale of what you consider to be a patch. And you need to define those patches. So in experimental systems such as this Metatron in Moulis, southern France, that was set with experimental units with little connecting corridors that you can close or open, then it's pretty clear what a patch would be. But for a natural landscape and some animal or plant, it's often really not obvious to decide what is a patch, what is not a patch. There are several types of patches that are embedded in each other. And there's also a lot of difficulty in the literature about dispersal, because those spatial scales are not made completely explicit in those different approaches. Then there's a very different approach, which focuses on dispersal distance, not only moving from one unit to another, but how far you go. So this is an example of experiment that we run with collaborators using those spider mites. And again, we had some discrete patches, which were little bits of leaf cuts connected by paraffin bridges. And we put some spider mites on this, and we record after a while where we find these mites and whether it has dispersed from one patch, two patch, four patch, and so on. And then you measure the distribution of mites that you found at different dispersal distances. Here measure the number of patches that they crossed. And you fit some dispersal kernel, and you can, so there the vision of dispersal is vision in some continuous space, a bit like the different type of models we also discussed before earlier this week. And what you want to know is what is the distribution of dispersal distances, and you may wonder about how the moment of this distribution are evolving. I mean, is the mean dispersal distance changing? Is the kurtosis, the skew of this distribution changing? And so it's not like a yes or no, dispersing or not dispersing. You've got more information about how far you went. So why is dispersal important? So it's a bit less interesting in the context of this course because we discussed that already a lot. There are many different reasons. There are purely demographic reasons, like dispersal capacities are going to be limiting spread rates in many circumstances. And demography of population, this is just one example of modeling the recommendation of Europe by common beach after last glaciation. And you can see that through time, using reconstruction of past climate and some niche model that I already discussed, if you model only the climatic niche of the species, the color code gives the probability of presence of the species through time, and the black dots are the fossil evidence of its presence across Europe in time. So of course, the fossil coverage is not perfect, but still, you predict that beach should be almost everywhere much sooner than it seems to be. And if you do a different type of modeling, integrating dispersal capacity of the species, you predict much better the actual distribution and spread rates of the species through time. And the conclusion of this study is that the natural spread rates of beach are too slow based on what this species was able to do in the past to cope with current climate change. So that's one reason why you are interested in dispersal, a purely demographic point of view. And also, it is important because it has huge evolutionary consequences. So I already mentioned the effect of dispersal on the evolution of species niche, and this is an experiment testing it with viruses. Show you something? So in this study, they had serial passages, and through these passages, so they experimentally adapt a phage to a new bacterial host. And they had phages in different amounts at each passage, and they assayed fitness after 30 passages. And what they found is that the fitness of the virus population after the 30 passages time of evolution on this new host was depending very much on the number of migrants that you were adding at each passage. So here as a control, you added also migrants, but so to be able to infect this new host, you have to have a mutation that the wild-type population don't have. So if you had migrants from this wild-type population that has no ability to infect this new host, you have no effect of migration. But if you infect with mutants that already have the ability to infect this new host but have a poor fitness on it, then you see that you maximize adaptation to this new environment for intermediate levels of migration. So too little migration is not good, but too much is not good either. So dispersal affects the evolution of the ecological niche. We've seen theoretical prediction about that, and there's experimental evidence for it as well. Dispersal through gene flow affects many other evolutionary processes, such as genetic divergence and speciation. And this is just one interesting study looking at how dispersal distance in birds might be correlated with degree of diversification in those groups of birds. They use a data set with about 70 species of birds for which the mean distance between the mean natural dispersal distance is known. And each of those species, there are different species that have been described. And what you see is that in species with short distance dispersal, there are more subspecies described than in species that disperse very far. So, of course, it's completely correlative, so it could be many other things. And this is the effect of phylogeny is corrected for in this analysis. But it seems indeed that restricted dispersal is somehow helping diversification and evolution along the way to speciation if the number of subspecies reflect that. So the second question in the review was, so is dispersal plastic? So can you define for me phenotypic plasticity? You know what we call phenotypic plasticity? Yeah, so in general, plasticity is the fact that the same genotype will express different phenotypes in different environments. So for dispersal, it means indeed that as you take individuals with the same genotype and they encounter different environmental conditions, they will have different dispersal behavior. So the answer is yes, and there are many evidence that dispersal distance or dispersal rates depend on many features and that dispersal is not the random diffusion process that we model all the time. The individuals leave their natural area and select a breeding habitat non-randomly. And there's been different terms in the literature calling condition-dependent dispersal or phenotype-dependent dispersal, and now what is fashionable is informed dispersal. And you've got a nice review that I think you had in your reading list, if you want to know more about that. And basically the idea that there are many external factors that might affect dispersal decision at each of these stages, like departure, tangent, and settlement, such as inbreeding risk in competition, intra-species competition, habitat quality, outbreeding risk, that those external factors affect the internal state of each individual, and that those decisions are going also to depend on the physiology, the behavior, the morphology, or the life history trait of those individuals. And that also what happens during transience and settlement might also feedback onto the departure decision of other individuals. So this idea of informed dispersal is that individuals gather and transfer information before and during their movement through the landscape. So one of those experiments suggesting that dispersal is informed, well, there are many experiments showing effects of many environmental conditions on dispersal behavior. I'm just showing one that is a bit intriguing. So those are dispersal assays in this protist called Tetrahymenae thermophila. So here's the kind of environment those individuals disperse into. They are just little tubes connected by little bridges with liquid medium, and those protists swim across those bridges and connect different tubes, and can stop connection by using these clips here, or you can open connection between tubes. And so in this experiment, what the authors manipulated is they used dispersal in one type of environment. They took the individuals that had dispersed in this environment and add them to another patch or tube, and witness how those few individuals that they had it from there to there affected the dispersal decision of the rest of the individuals. So for instance, what they did is that they had individuals that had dispersed from a poor environment into some empty tube, some individuals that had dispersed from a rich environment to some empty tube, and when you introduce these immigrants in a new population, you record how many of that population dispersed if the immigrants were coming from initially a poor environment or a rich environment. And interestingly, they find an effect of those very few individuals that were coming from different environments. Like if you look at the dispersal rates of those population, if they receive immigrants from a poor environment, from a rich environment, from an environment with the same quality, you have significant differences in dispersal rate depending on the origin of those immigrants. But this response is completely idiosyncratic. So this is what happened for line Q. If you take another line of Tetrahymenae, you do the same experiment, you get a completely different answer. So Conclusion for me is that indeed they found that dispersal is modified by the presence of immigrants coming from a different environment, and they're really excited about that because they think that there's really information conveyed about the nature of the environment by individuals that move across the landscape. But this is highly idiosyncratic, and it's not very clear for me what is the information that is conveyed about the environment. Yeah. No, I don't remember. The graph that I showed, it's for the same. Yeah, it was very small. Yeah, they found that repeatedly, actually. They have other experiments showing that. So they found that with other organisms like lizards, and so which maybe is less surprising that there's something that. But they found that with tetramina repeatedly, so it's not just an artifact. I think maybe, but but yeah, check the paper. It's not. To me, it's not completely clear. But the fraction that they introduce here is really small, so it cannot just it's not those same individuals dispersing again. It's something else happening. So I didn't have time to show you, but there's a nice experiment that those guys did recently, where they use that system to look at adaptation in heterogeneous habitats, and the fact that you've got this type of informed dispersal decisions is actually accelerating the evolution of local adaptation rather than if they force migration to be random. So, third question is, how fast can dispersal evolve? Because dispersal is important. It is highly influenced by the variation in the environment, but maybe it's evolving very slowly, and we don't need to care about dispersal evolution if we want to understand the impact of dispersal on many processes. So the best evidence for very fast evolution of dispersal comes from a natural situation of biological invasion. And since I wrote the review, there were really a lot of very nice papers about this system of cane toads that were introduced to control some insect eating the cane in Australia. They never controlled the pest, but they became a pest themselves. And nobody eats them because they are toxic, and they spread throughout the north of Australia and causing a lot of ecological problems. But because it's a recent invasion, there's very, very good data about where the toads were when they arrived, about all the spread rate and the demography, and they know a lot of very nice data about other aspects. So the cane toads were introduced to Australia in 1935. Means that there's been approximately about 80 generations of continuous special expansion in that species. And if you collect toads at the front of the range and in places where they've been introduced for a longer time, find a lot of heritable differences between the toads at the front and at the core of the range, concerning many traits like life history traits like larval growth rate, immune function, dispersal rate, movement strategy and morphology. And what they actually found is that because of the evolution of higher dispersal capacity and bigger legs and different behavior, the front are moving much faster than the toads are at the core of the range. And because of that, this fast evolution of dispersal at the edge has accelerated the invasion, and spread rate of the invasion has increased from 10 to 15 kilometers per year after introduction to 60 kilometers per year in 2006. It's a huge effect on the demography, which is completely mediated by the evolution of dispersal. So the mechanism of this fast evolution of dispersal at the front of the age is a bit trivial, actually. So it's called spatial sorting, but the mechanism of that is very simple. If you have some genetic variation in the ability to disperse and the dispersal distance, the genotypes that you're going to find most likely at the front of an expanding age are those who get there. And those who get there are those who have a genetic tendency to disperse more. So you've got a sorting through space that is just natural. It's just very simple. other genotypes that disperse further that you will find further at the edge. Because of this spatial segregation, those moving faster are at the location that determines the expansion of the population. Because the speed of expansion is depending on the speed of movement at the edge. Okay? So more generally, if you think about the evolution of dispersal, you need to think about what are the phenotypic traits, because, okay, dispersal by definition is a movement, but, okay, so you can measure heritability for a movement, but you know that this movement is the result of some behavior and some morphology and some ability or some motivation to live. So to understand the evolution of dispersal, you have to understand what are the phenotypes that are affecting dispersal and how they evolve, are they genetically variable, are they constrained by different genetic constraints. So there's been historically a lot of work on species where there's strong, obvious dimorphic types affecting dispersal. So for instance, in many asteraceae plants, the dispersal of seeds is affected by a parachute-like structure, which is called a pappus, on the top of the seed. And different species, or even within the species, you can have variation in the size or architecture of the pappus. And in some species that are called heteromorphic, the same plant produces seeds with a pappus, so with a dispersal structure, and seeds without any pappus, without any dispersal structure. So within the same plant, you have two types of offspring that obviously have very different dispersal distance. In insects, you will also have a lot of work on wing dimorphism, where again, in the same zip shape, you can find offspring with fully developed wings and offspring with generally a big abdomen and very high fecundity, but no wings or very short, aborted wings. So in this case, you know, it's kind of easy to say, okay, one proxy for the dispersal rate is going to be the proportion of seeds with a dispersal structure or insect with wing rather than with no wing. It's not always the case that it's a good predictor. For instance, in this species here, which is Centaurea corambosa, you have a big pappus, but it's completely useless. It's like, you know, a parachute where you had removed the cloth. It's just like an umbrella with no cover. So actually, those seeds are not dispersed far at all, despite this structure. This structure is used by ants to drag the seed to their anthill. It's not. It's useless. And so you also have insects where insects have wings, but they don't fly. So you have to be a little bit cautious about that. There were also studies looking at less obvious aspects of phenotypes that could affect dispersal. So this is a very nice study in Arabidopsis thaliana made by Kathleen Donoghue and a group. And here they had a lot of different genotypes of Arabidopsis. And they recorded for a lot of individuals and genotypes the whole dispersal distance distribution of seeds. So the distance, the standard deviation of distance, the kurtosis of the distribution, and they correlated those parameters of the dispersal kernels to the architecture of the plant, seed and plant traits, both at high density in growing conditions such as with high density of plant or low density of plant. And you see that, for instance, high density of plant and very few traits that correlate with dispersal distance and many more so at low density. So this genetic or phenotypic architecture of dispersal is also depending on the environment. Okay. There's also one example that became very famous. It's an example of a major gene in a butterfly, in this butterfly Melitia kingsia, which is well known because it's a butterfly that Ilkka Anski studied in a very famous metapopulation in Finland for years and years, and essentially doing ecology at the beginning, but they started to look at being interested in the evolution of dispersal in this system, and they found variation in a single gene called PGI, where this variation, where different genotypes are associated with lots of different traits associated with metabolism, but also behavior, movement, life history, and seems to be a major factor driving variation in dispersal distances and behavior in that species. These examples here I wanted to show you to show that, so again, in Arabidopsis thaliana, recording dispersal distance, average dispersal distance for different ecotypes of the species. They found a lot of variation among ecotypes. They found a big effect of the environment on dispersal distance, so a big plasticity effect here, as shown because dispersal distance is higher at low density than at high density, but very few genotype by environment interaction, meaning that all the ecotype responded to density about the same way. So if you've got genetic variation for those traits, and dispersal potentially can evolve fast in many situations. Now, dispersal has been, so to try to understand the evolution of dispersal, been a lot of theoretical work. And this theoretical work was based on different models with different assumptions. So people got interested in the evolution of this trait very, very early on when we didn't have very fast and powerful computers. And what people had to do, try to understand that, is basically write a mathematical model that you could solve, and you could get answers about the evolution of dispersal based on this model. So to allow mathematical tractability, those models in general made a number of very strong assumptions about the nature of space or the nature of dispersal and so on. So I've listed a few of these strong assumptions that so often most of those models assume discrete patches of habitat. There was very little work looking at some kind of continuous space. Those models generally assume either very few patches, like 2, or an infinite number of patches, which is another interesting mathematical limit to try to drive solution. And they also assume either very few individuals per patch, like typically 1, or an infinite number of individuals per patch, basically neglecting all stochastic components and having a deterministic model. Often space is described only implicitly. I will explain later what it means, meaning that all patches actually were connected to each other equally, which again is not completely what happens in reality. And if they were going into more sophisticated description of space and connectivity, often at some point they need to neglect higher moments of spatial interactions to make progress in terms of analysis. I just wanted you to point you at some analytical approach that was developed relatively recently that is still mathematical, but relax a lot of this. It's a spatial, the space is continuous, some variation in habitat quality that is also continuous with some kernel describing how patches are formed in the landscape and autocorrelation, spatial autocorrelation of spatial qualities through space. And then there are some kernel describing how competitive interaction decrease through space, and some dispersal kernel, and some mating kernel, and using this different approximation about the parameter in the model, they managed to close the problem of moments in spatial interaction closure. So it's a promising technique. The problem is that it's still very complicated, so it's not clear that you end up with closed form solution for what you're interested in. So now, what is the most popular approach to try to understand the evolution of dispersal is just to run massive, big computer simulation where you describe spatially explicit populations. It's not really costly to do anymore, so you can have an example of, so it's called spatially explicit interval-based simulations. You don't have to make assumption about finite population size, you don't have to make assumption about interaction being global or local. You just simulate a bunch of individuals that are moving through space and you get answers. This is just one example of some expanding species. And so they recorded the joint evolution of 3 traits, which is reproduction, dispersal, and competitive ability. through space, and here you've got 3 snapshots of those populations through time. And what you see is this population is expanding. You see that the dispersal genotypes in dark here are located at the front of the range. At the front of the range, you also have individuals with lower competitive ability, but reproduction is not so clear that you see a pattern. So a lot, if not most, of recent research about evolution of dispersal from a theoretical point of view is doing these kind of things. However, you have to be aware that every model, even if you do a spatially explicit simulation that looks just like reality, makes assumptions. And if you do a very complicated model with spatially explicit space and individuals that do lots of things, you make a lot of assumptions. And then it's really hard to understand what are the mechanisms at stake that explain the result you obtain, and which are the assumptions that you made that really matter for the result you obtain. Sometimes you make an assumption just because you didn't think about some alternative idea, but you didn't really want to, it's not really important for you that individuals behave this way. And it has a huge impact on your result, and you don't know about it. So recently, there's been a lot of debate between 2 groups of people that are doing a lot of those simulations about the evolution of dispersal and how it should depend on density, and also, again, inform dispersal. And those 2 groups were obtaining results that were really different, and it was really not clear why they would, because their models looked really similar. And so they argued, yeah, I'm right, you're wrong, and so on. But nobody knows really what's going on. And that's, to me, a big warning. When you do this kind of simulations, it's always good to have some analytical, simple model you can compare your simulation to. And it's good to understand what is going on in your model. And it's not always the case in those big simulations. And sometimes, also, putting a lot of realism into the simulation is not going to teach you more than a simpler model. Yeah. 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 There were no analytical work on that, but you could model it this way, except like here you've got this stochasticity and you have 3 traits evolving at the same time. I'm not sure that has been modeled analytically either. But, it's just it's a spread of, it's a population that is expanding through space. Is that what you want to know? Or? Oh yeah, I don't know about that. So this is why I think it's still useful to look at those old models that were analytical models and very simple with silly assumptions, because I think we learned a lot through them. And it's okay for me to run very complicated simulation, but it's good to build on what we know already. And those can be really useful to understand what will be going on in more complex models. So the issue that those modeling approaches faced at that time is which aspect of space we want really to keep in the model because we think they are important, and which aspect of space we don't want to keep in because it complicates our mathematical work too much and we can't, and maybe they're not that important for the process that we want to model. So one model that was really used a lot in population genetics and also in evolutionary ecology was the island model of migration. And basically the idea is that we want to, the way we're gonna think about space is that we want to distinguish local interactions and global scale. So we only have those 2 scales, something that is happening at the global scale and something happening at the local scale. And we don't really care about intermediate kind of scale. So what is this model assuming? It's assuming typically that you've got a number of population or sites throughout the landscape. These sites can be occupied by a population or can be empty. And occupied sites, there's some demography within those sites. They produce some offspring, and a fraction of this offspring is going to remain in the natal patch, and a fraction of this offspring will join a big dispersal pool where the dispersing individuals from everywhere in the landscape are going to be mixed. And then this big pool of disperser are going to be distributed randomly across the landscape. A fraction of them will actually die in the process and land in the sea if you want. That's why if you want to pursue the island analog or paradigm. So a fraction of those dispersing individuals will never make it to anywhere. And a fraction 1 minus c, so fraction c will die, a fraction 1 minus c will be distributed completely at random across all occupied sites. Okay, so it's not such a bad model, like for instance for organisms that disperse very widely their seeds or pollen, like for instance in some tree species, it actually works quite well sometimes to describe dispersal. But obviously it's not how things work in general, but still, I mean, with this system, you have a local scale, and globally, then you have all of the patch, which is a global scale. Then early on, also in the history of population genetics, they were also, well, did you see, you discuss this kind of models already for coalescence? Okay. So another alternative model that was developed early on in population genetics is a stepping stone model. So here you have more notion of space than in this other model, because what you assume is that, so you've got, for instance, patches that are arranged like a little path on the line, and you can disperse, so you can either disperse only to your nearest neighbor, or you can have some distribution of dispersal distances, so you can disperse either 1 patch away, or 2 patch away, or 3 patch away. And then you're going to look at the distribution of those dispersal distances. So of course, this linear arrangement of population, which is, by the way, what we also assume in the model of species range we discussed, is not very general. In general, space has at least 2 dimensions. And so you can have the same kind of stepping stone model. And often what people do, because they don't want to have edge effect, which is complicating the analysis, they assume some kind of torus situation where actually you have no edge, because if you disperse somewhere, you always have neighbors. So it's, again, some kind of very theoretical idea of space that works to obtain mathematical model derivation. And then there's also a lot of models that are even more extreme than that, that assume, so instead, so yeah, what I forgot to say is that often, here you also assume an infinite number of such patches. And this is also potentially the case with those models. And at the extreme, there are also dispersal models that were developed in the 90s and 80s, where you assume just 2 patches of habitat. And you look at dispersal between habitats of different quality, and you'll see some examples of that. And despite the simplicity of those models, they were also very useful in terms of thinking about what's going on with dispersal. Then there's been also some attempts that have not really been used so much, but are interesting, to look at random network models to describe dispersal. So here's the idea that a patch is connected to some other patch, so it's not equally connected to all the patches in the met population. And you can disperse if some patch is freed by the death of individuals next to you, for instance, but you cannot disperse to an empty patch somewhere else. And then you look at the demography and the evolution at this scale of interaction. But this is a random network because any patch is connected to a number of networks, but the whole network structure is not kept into the model, basically. You don't really have information about who is next to you. You know who is next to you, but you don't know who is 2 steps away from you. Okay, so start that, and we'll probably have time, won't have time to finish, but I'm yeah, we'll have time to finish. So just to help you, if you come across some of these models to get an idea of how those works, there's more complicated version of that, and if you are interested in really the most rigorous and really connected to a lot of concepts in population genetics approach with complex demography and so on, there's a paper that we wrote with François Rousset, which I think there are a lot of things. I warn you that if you read that, you will have an headache, as I do every time I try to reread what I did. But it's quite useful to connect to a lot of population genetics models, this idea of evolution of dispersal. So I will do a simpler version for you now. So if we want to model selection on dispersal, we're going to do what every population genetic model does. We're going to assume that we've got some allele M. So we're going to work with some haploid model of population genetics. So we've got some allele M, and so we assume that individuals with this allele produce offspring with a dispersal rate C. And there's a modifier dispersal, so an little m, which has a slightly different dispersal rate. And what we want to do is to look at, so if we want to predict change in allelic frequencies, we want to look at the fitness of this allele M, so having the phenotype Z star. And typically, in models of dispersal evolution, this fitness is dependent on what the other genotypes are doing in the population. So there's not a fitness attached to one genotype. The fitness depends on who you are competing with. So that's what I write like that. So this is the fitness of genotypes having dispersal rate Z star when they compete and when the dominant strategy and the most frequent strategy in the population is Z. So basically, the whole idea is that we want to know how dispersal rate evolves. We want to know whether mutants, so imagine that we've got this allele, we want to know if this allele is going to be invaded by mutants that have a higher dispersal rate than the resident, or mutants, or will be invaded by mutants that have a lower dispersal rate than the resident, or whether they cannot be invaded by anybody. And then this is a definition of, did you see that at all during this course, the game theory kind of stuff? Okay, so, well, first of all, we want to know what type of alleles are able to invade this population. And this will be given by the condition that I wrote up there. So if this is positive, okay, it means that the fitness if this is positive, it means that this has a better fitness than the resident. So you will have selection favoring mutant with a positive delta, so increasing their dispersal. And if it's negative, then the mutants that are going to invade are those with a negative delta, so a lower dispersal rate. And there's another situation. Which is that maybe this allele has a dispersal rate that does not allow any mutant to invade. So no mutant has a better fitness than this strategy, when this strategy is dominant. And this is what we call an evolutionary stable strategy, because if you had such a genotype and such a that associated with a particular phenotype, then it's resistant to invasion by any mutant that you can consider. So you expect, if you come to see, to study a population, you expect to find genotypes with this strategy in the population, or the most frequent genotypes should be in that strategy. So those will be defined by the fact that the fitness sorry. And the second derivative has to be negative for it to be a fitness maximum. So this is very general, and it could be in any trait, okay? So what we do then is, so we want to compute the fitness of, so first we have to define fitness. So let's say fitness, here we consider it's a number of surviving offspring of some individuals with some genotype. What we're going to do is pick up a focal individual, and we will try to compute what is the expected number of surviving offspring of that individual. So we call Z dot, just for simplicity here, the dispersal rate is a fraction of your offspring you're going to disperse. Okay? Not what you do yourself, it's what your offspring do. Since it's an haploid model, it's a bit the same. So then you compute the fitness of this focal individual, given that you've got some dominant phenotype in the population. So let's assume F is the fecundity. The number of offspring everybody produces. Then a fraction 1 minus Z dot are going to stay in the natal patch. And those will have some survival, which I call W philo, like Philopatrick. And the other offspring are going to disperse. A fraction of them are going to die in the dispersal process. And on average, the survival will be W disp, which is the survival of offspring somewhere else than in the natal patch. That's really simple. Now, if I want to compute this derivative, so the first thing is that if I take the derivative with respect to delta, assuming that my focal intervals has a little m, if I take the derivative of this bit here, what do I obtain? I derive this bit with respect to delta. You remember when you have when you take the derivative of a product, what is the derivative of a product? Well, so first of all, I always think, so it's always wrong, but so those who should not be partial derivative, but everywhere should be d. Okay. If I derive a product xy, okay, do you remember that? So if I take the derivative of this one, okay, let's write it. So here, I assume that my focal individual has this, has a little m. So if I take the derivative with respect to delta of this quantity, what do I get? I can't hear you. I've got 1 minus that dot, just minus 1. So I got minus 1. Then if I take this derivative, what is it? Plus 1. Okay, so then we get to rearrange things a little bit. You see here you've got this minus the survival probability of Fitzpatrick individuals, plus y minus c is the survival of dispersing individuals, which is actually the survival of dispersing individuals at birth, because then there's the survival during dispersal and the survival once you immigrate somewhere. So if you look at this selection gradient, the first part of it is just simply the difference in survival between Fitzpatrick and dispersing individuals. It's pretty trivial, okay? So you can expect that if you had to ignore this, well, simply you will get higher evolution of higher dispersal rate if the survival of dispersing individuals is greater than the survival of Fitzpatrick individuals. Very, very trivial, okay? If you survive better by dispersing, then genotypes that invest more into dispersal should be advantageous. And the reverse is true. If the survival in the home site is greater than the survival when you disperse, for instance, if this survival rate during dispersal is very low, then it's better to stay at home. And yeah. It's not here, because so Z dot is a general, you will see that later, maybe it's just confusing. Z dot is just a focal individual that you consider. But here you're considering a focal individual with the mutant allele, because that's what you want to compute to know whether the resident strategy is going to be invaded. Okay? But this equation, this equation is true for any genotype. Okay? It's just that here, yeah, what I should say, here I take this derivative for a focal individual with a little m allele. Okay? So this first part, I mean, it's part of every model of dispersal evolution. If on average your survival is greater if you stay or if you go, then you evolve more or less dispersal. But what you can see very clearly is that if there were only those terms, and if those quantities, like the survival of Fitzpatrick individuals and the survival of dispersing individuals were constant, then what would be the AESS strategy? What would be the only possible evolutionary stable strategy? If this is constant, is this constant as well with different values? Like the sum of this term is going to be either negative or positive, so which means that what happens? If this is negative, for instance, you start with a strategy with some dispersal rate, and the selection gradient is negative. What will happen to the dispersal rate in the population? It will go down. But it will always go down because it does not depend on what the dominant strategy does. It will always select for lower dispersal because they just have a better fitness. So the only ESS strategy would be 0 for the dispersal rate. Conversely, if this is positive, you have a selection gradient that does not, and this selection gradient does not change with the strategy, then you will always have increasing dispersal. Well, the difficulty comes now that those survival in general are not constant. They depend on what the other strategy is doing. So you could also ignore that, but this is not constant, and actually it could be value of the resident strategy seed for which this is positive, and there will be value for which it's negative. And then you might be able to find the ESS strategy for which this quantity is just 0. Now, what are those other terms? Those other terms, they exist if the fact that, so if the mean fitness of a Fitzpatrick offspring is depending on the strategy of my mutant. Okay? Otherwise, its derivative is 0. So it means that the survival of a Fitzpatrick offspring is depending on the strategy of the mutant. And the same here. This term is not 0 if the mean survival of a dispersing offspring is depending on the phenotype of my mutant. So when will that happen? compute, well, again, I'm always using the wrong G. So this means the expected survival of a Fitzpatrick offspring will depend on the number of, typically on the number of individuals that are competing with these individuals in the same patch. So what it depends on is, and so those, this number of individuals you will be competing with depends on how many individuals that were born in this patch stayed in this patch, which depends on the dispersal rate of all other offspring, of all other individuals in the same patch where you were born. And it depends on how many immigrants arrived in your patch, which depends on the average dispersal rate in all other patches in the landscape. And so if I write as Z0 the average dispersal rate of individuals in the natural patch, and Z1 as the same in other patches. Then using the chain rule, I can say, okay, my survival as a Fitzpatrick offspring is depending on the dispersal rate in my home patch, and how this dispersal rate in my home patch depends on my genotype. It also depends on the number of immigrants, which depends on the dispersal rate of other patches, and how this dispersal rate of other patches is depending on my genotype. So all this is a little bizarre because you want to, I mean, okay, this is the genotype of the focal individual. Why is there a relationship between the genotype of these individuals and the phenotype of other individuals in the same patch? The reason why you have this, so if you compute this average dispersal rate of individuals in the same patch as you, so if I draw some other individual in the same patch as you, these individuals may have, may carry a different allele, may have a different genotype than you have, or it can have the same genotype as you have. Okay? So in the same patch, you might have individuals that are sharing the same genotype and are identical by descent because of relatedness, because the history of this population. And so with probability 1 minus Q0, the other individuals that you draw is not carrying the same gene as you, so it's carrying the big M allele, and then its dispersal rate is going to be Z. And with probability Q0, it's going to carry the same allele as you, which means that its dispersal rate will be Z plus delta. Which means that the average dispersal rate in your home patch could be written something like that. So here this term can be replaced by Q0, and here Q1, would be the probability to draw in the same population as you, and individuals with the same gene. And here's the probability to draw in some other patch than your patch, an individual with the same gene. So just to come back to this expression, in many models of evolution of dispersal, you've got this kind of terms, which is just a difference in survival between Fitzpatrick and dispersing individuals. And anytime you find some kind of genetic structure or phenotypic structure, so that actually there's some probability that in the same patch there are individuals with the same gene as you. And so you've got selection on this focal individual, which is indirect through the correlation between its own phenotype with the phenotype of its neighbors. This is an indirect effect of genotype on those survivors. And this will occur anytime you've got any type of genetic structure in the population, and anytime the probability that you find individuals that are resembling you is higher if they are in the same population than in some other population. So basically, then you could have lots of different assumptions about, exact function, how the survival of Fitzpatrick or dispersing individuals depend on the mean phenotype. You can add a lot of complexity and stuff in there. But this is a basic approach to many, many of those models. And so you can solve those models by classical population genetics models, sometimes multi-locus models. You might see some examples of those. You can use adaptive dynamics, which is basically what I'm doing here, trying to look for equilibrium in the long term. But both of those approaches, most of the literature about dispersal evolution is considering long-term evolution equilibrium and not really looking at the dynamics of evolution of dispersal. And it's only quite recently, motivated again by this kind of transient processes such as invasion processes, that there were some models that were actually short-term quantitative genetics models of dispersal evolution. And which I find really interesting because in many applied contexts, so we understood a lot about the evolutionary forces acting on dispersal through those long-term evolutionary equilibria, but those are not really so relevant to understand the dynamics that are affecting those processes of recolonization or invasion or stuff like that. So here in this model, again, they looked at the invasion of northern Australia by cane toads, and they contrasted the spread rate under different scenarios using estimates of heritability of dispersal behavior in those species. And you can see that so they had dispersal with scenarios with no evolution at all, or scenarios with only evolution of life history but not dispersal, evolution of dispersal only, or evolution of both dispersal and life history. And basically the conclusion is that if you assume spread without any evolution, in 72 years, the species will have spread by 470 kilometers. If you've got evolution of both life history and dispersal, they actually spread by 1,000 kilometers. So it's not a trivial effect at all. And then it's not only, you can understand why migration will be bigger at the front, but having quantitative prediction about how fast dispersal will evolve in the system is really important. And also something that is interesting in this example is that so both the evolution of life history and dispersal are accelerating the spread rate of that species, but the spread rate is even bigger if you have two together, and it's bigger than just adding the separate and additive effect of evolution of dispersal or evolution of life history. Okay, so the sixth question was, does dispersal allow escape from competition, and more precisely about escape from kin competition? So I will first report about an experiment that Susan Keltz did in the late 90s. she worked on this plant called Trillium grandiflorum, which has seeds that have some structure which is called an elaiosome, which is a little pocket of nutrients that ants really love. And the ants will take the seeds and generally carry them to the anthill and eat the elaiosome. And in the process of doing that, they lost a lot of seeds in the landscape and they disperse the seeds. So this elaiosome is understood as a structure that favors dispersal by the ant. So in this experiment, they collected seeds on plants, they radiolabeled the seeds, and they use a code tag to mark different kinship, so different pools of seeds formed from the same mother plant. And then they put back the seeds just at the bottom of the plant, and they let the ants come and do their work. So they had little, piles of seeds below each mother plant, and they just came back after the ants had done their job, and they recorded the position of every seed in the landscape. And what they found, they did the experiment twice, and what they found is that they found decreased aggregation of seeds after dispersal of seeds. So if the seeds had just fallen beneath the plant, they will be all packed together, very high density, and because of dispersal by the ants, now the density of seedlings around each seed was on average smaller. So they found that just one year, and the other year it was not true. So it's not obvious always that dispersal decreased aggregation, because also the ants could also aggregate even more the seeds because they form little piles of them. But this gave support to the idea that was in the literature for a very long time, that indeed dispersal evolves because it allows to reduce competition between seedlings. So there have been a number of models that have tried to test this idea, that dispersal is a way to avoid crowded conditions, and it's selected for because it reduces competition. And early models that look at habitat heterogeneity in density actually, interestingly, found just the reverse. So what they assume is that you had patches of habitat where the plants could grow, for instance. And so you had some habitat that were with more resources and with a higher density of plant, and bad patches was a lower carrying capacity, less resource, so less plants could grow in those patches. And if you assume that this kind of heterogeneity in habitat quality is completely stable, what you find is that, what you find is that dispersing genotypes have always a disadvantage in this environment, and you select for zero dispersal in this model, which is very bizarre. And the reason why is that, for that to occur, is that so you've got this fixed difference in habitat quality. So because here you've got much more individuals than here, you have a net flow of individuals from this habitat to this habitat. The dispersal rate is the same. Every individual has the same probability to move. But as we've seen before when we discussed sourcing models, if the dispersal rate is constant, then a large proportion of those individuals, well, the same proportion, but a higher number of individuals will move from this rich, good quality habitat to this poor habitat, and much less in the other direction. So dispersing genotype is ending up leaving very often good patch of habitat to end up in very poor quality habitat. And a non-dispersing genotype will increase in frequency in those good patch of habitat because the dispersing genotype is leaving and not returning. So if you assume passive dispersal completely tied to the quality of habitat and these fixed differences in habitat quality, you find that you lose dispersal. So this is also assuming, this model is also assuming a very large population size, so all these indirect effects here are completely negative. So it would not be the case if you were taking into consideration the genetic structure of patches. Now, if you assume that you have such habitat heterogeneity, but it's variable in time. So it's not always the case that this patch is the best patch every year, and this patch is the bad patch every year. You've got fluctuation in carrying capacity through time in any patch. So this patch can become a bad patch next year, and this patch can become a good patch next year. And if you do that, then you find that dispersing genotypes have an advantage. Because then, in this case, indeed, there will be a given year a net flow of individuals from good patches to bad patches. And if these bad patches with very low density of individuals becomes a good patch next year, there will be a lot of room, and there will be very little competition in this patch. Because the local plants that were there were not very numerous, and so they will produce a few offspring. And so the genotype that is dispersing from the good patch to the bad patch has an increase, has access to more resources and less competition than the reverse. And so the reason why you select the dispersing genotype in this system is because they avoid more often crowded conditions than the genotypes that stay where they are and are always very numerous in patches that were good quality the year before, but might not be of good quality the next year. So an extreme example of that is when you've got catastrophic extinction. So it's not just that the patches are varying in quality and carrying capacity from year to year, just that you have a fire or a frost or anything that just wipes out every individual in the patch, and this patch is empty. So a lot of models like that, metapopulation models, including the first one by Van Valen in the 70s, modeled the evolution of dispersal in such a landscape. So here you've got many, many patches. You've got catastrophes that are killing everybody, so those patches are empty and can only be recognized by dispersing individuals. Then it's quite obvious that those empty patches with a lot of resources that is not used can be colonized only by dispersing genotypes, and this creates some selection for dispersal. And actually, those models found, all those models found that the ESS dispersal rate, so the strategy here that would result in no mutant being able to invade, is such that the dispersal rate of this strategy is just equal to the extinction rate of patches. This is true if the cost of dispersal is very high, if you've got very large population size, so you can neglect all this genetic structure, and also if you've got very fast population growth, such that after an extinction, you've got recolonization so fast that you already reach your carrying capacity the year after. But this is the same explanation. Here in these models, dispersal is selected for because it allows you to move from a crowded, fully occupied patch to a patch that is empty with a lot of resources. So those models assumed constant fixed dispersal rates, not informed by anything. And already in the model by McPeak et al. In 1992, when they found that they could not select for dispersal, they relaxed this assumption and looked at whether you could still select for dispersal if dispersal was a plastic strategy. What they found is that, indeed, if dispersal was different, if you were born in a patch of poor quality or bad quality, so if you could adjust your dispersal behavior depending on the quality of habitat in which you were born, then you found very different results. And what they found is that the ESS was defined by the fact that you exchange an equal number of migrants between the two patches. So it's not the dispersal, so it means that this time, previously the dispersal proportion was fixed, but the number of migrants exchanging between patches was different, and you find that an equilibrium is reached when the number of migrants is equal, which means that the proportion of dispersal is different. So if this patch is sending the same number of migrants in this direction, then this big patch is sending migrants in this direction. I mean, here the dispersal rate, the dispersal proportion per individual is much higher than here. And it makes sense. You disperse out of bad quality habitat, and you disperse less when you're good quality habitat. So it's very intuitive. It's also related to the idea of ideal tree distribution. I don't know if you heard about that before. How you forage to space and how you allocate or distribute your density across space depending on resource availability. So this is true in some stable habitat. So then you can have some dispersal if individuals actually know something about the habitat where they're living. In unstable habitats, there's been a lot of work since then looking at how dispersal probability should change with population density in your natal patch. And often those models find things like that. There's kind of critical density below which you disperse none of your offspring. And once you reach that density of population, you start to disperse more and more of your offspring to other patches. So the fact that you expect dispersal to change with population density is something that has been predicted many times by models. So we'll just finish with this empirical example, and then tomorrow we'll continue with the rest. So data about density-dependent dispersal, however, is much more complex than what the model predicts. So for instance, this experiment done with, in this common lizard called Lacerta vivipara. Actually, the name has changed. I think it's Zootecana vivipara. So those are experiments done by Jean Claubert and his team, where they had experimental settings with patches with population of lizards connected by little corridors, and you could close those corridors, and there were traps, so you could capture every individual attempting to disperse in one direction or another, and you could frustrate dispersal by closing the corridor. And what they found is that, so they looked at individuals dispersing from one patch, joining another patch, and they look at if those individuals would actually say, I don't like it here, I want to go back where I was. So they would capture them going back. So this is what is indicated here, is the probability of leaving the arrival population. So those are dispersing individuals that finally didn't want to go there and wanted to return to their home patch. And they look at this probability depending on the density in their patch of origin and the density in the patch where they had attempted to disperse. So if those individuals that dispersed from a population that was initially of high density, it did not make a difference for them if they arrived in a population with also a high density or a population with a low density. This is not significant. If the population that dispersed out of a population with a low density, then those arriving in a population with high density stayed, but those that had escaped from a low density population and found another population with low density were really unhappy about it and returned to their home patch. So density is something complicated and also shown by the previous model. Density can be a cue for the level of competition you have in some environment, but high density can also mean this is a good environment compared to a very low density population where you say, something is wrong here. If you get into a restaurant and nobody is eating there, generally you don't want to stay in the restaurant. And it's not because you are afraid that you will not be served quickly enough, is that you say, maybe if people have not come here, it's not a good place. So animals do that also pretty often. And sometimes they feel, it's too crowded, let's go to some other places. Or they feel, there's nobody, let's go to some other place. And the individuals that disperse at high and low density are not the same individuals. And they also have a lot of experiments showing that the personality of those animals that disperse at low and high density are different. They have different levels of aggressiveness, different levels of exploratory behavior, and also they have different abilities to colonize a new population. So the relationship between competition, density, and dispersal is probably still much more complex than what the models are exploring at the moment. Okay, the final part tomorrow. Do you have questions? Coffee.